Hi guys, welcome to Learn Extra Live, and we're doing life sciences today. And I'm John, and this is Kathy. And
hi guys.
And today for grade 10s, welcome. I'm not usually here, I usually sit in the background, but Indiana, we've had a little bit of a problem and she couldn't be here, so I'm stepping in to talk to you. Today, what are we doing, Kathy?
Well, people, we're going through mitosis again, because remember last week we did mitosis. We're going to finish mitosis off because you have to know this section very well, all right? We've gone through the phases, the first 3 phases, and the overall process. So, John, that's what we're going to be doing. We're going to get the learners to know mitosis, that they never, ever have to study it again. All right, guys?
Right, well, thank you so much, Kathy. Let's just go to a break, and after the break, we'll be back. Remember to get onto Facebook, post your questions, and I'll be chatting to you on Facebook and on Twitter, and let's get going. Let's get on with this lesson. See you just now. Welcome back, everyone. Hi, I'm John, and this is Learn Extra Live, and we're doing life sciences today. Now, we've got Kathy, and she's going to be looking at cell division today. We're going into mitosis. So please get onto the page, get onto the Facebook page, phone us, and we'd love to hear from you. So, Kathy, please take it away.
Thanks, John. Okay, people, just to recap from last week, we did cell division. Now, what kind of cell division? We are doing let's just get our colors right here while we're at it. Let's go for a nice bright yellow. We're doing mitosis. And we said that when my toes grow, okay, my shoes get too small for me. Mitosis. And it's for, remember the 4 reasons why we have mitosis. We have it for growth. Grow, nails, hair, in height, in width, get fat, lose weight, pick up muscle, do gym, etc. That's growth. For repair, so if a cell gets damaged, for replacement of any cells that are so damaged they can't be repaired, and then of course reproduction, but in unicellular organisms. In other words, they only have one cell. So unicellular organisms, they don't have reproductive organs. It's just one little organism, one little cell. So it has to make, if it wants to make more of its own, it has to make another one of itself. And we call that binary fission. Remember? Binary fission. All right, it's a form of mitosis, and it is for reproduction. Now we have our different phases, and the different phases are within the whole process. Now, during cell division, we have interphase, prophase, metaphase, anaphase, and telophase. Remember that interphase, I'm going to put in 2 little inverted commas there, interphase isn't really a phase. It's the phase before the phases start. That's why we call it interphase. But it is a time period. It does take a long time because interphase is like your preparation phase, like if you're going to have a birthday party. 3-4 weeks of preparing, and then the party's 3-4-5 hours long, but you've prepared for a long time to make everything happen and be successful. So interphase is, remember the word inter means between, all right? Intra, T-R-A, is inside, inter is between, all right? So we have the interphase, which is the preparation phase, and then we have the whole process. So the cycle starts at this point here, marked X, and it goes all the way around here. The cell grows, and it gets ready, and it develops all the way around here, and everything starts to now come in place, And then we have the G2 phase where the cell's now getting chemically ready, and we go into the M part, which is the cell division where we have prophase, metaphase, anaphase, and telophase that will then take place. All right, people, just remember, interphase is not really a phase. And when we see interphase in a slide or with a whole group of cells, you can't even see what's going on. It just looks like a normal cell. Other than the fact, or except for the fact that the nucleus looks quite large. So you can identify the nucleus, and that says, this cell's getting ready to undergo mitosis. And also something else, remember that mitosis will also, for growth, definitely take place in the growing tips of plants. So the little tippy, called the apical meristem of the bud, all right, and at the roots, right at the tip of the roots, those are growing points. And if you were ever asked, and if you have the proper equipment at school, and your teacher asks you to do slides of mitosis, you would take the tip of a root, because it's the cells there are actually quite large, and you would then take that, use a scalpel, a very, very sharp scalpel, cut it, make a slide for yourself, prepare the slide, and then have a look at it. But what they'll probably do is give you micrographs, which is very nice. So your teacher could give you a micrograph and say, right, what are the different types of mitosis taking place, the different phases, and we're going to go through that too. So just recapping again, remember, so here's our interphase. It is the between phase. Now, please never call it a between phase in an exam. I think I will die. So don't you dare do that. But it's to explain to you that's what it is. It's an in-between phase. Every time mitosis is going to take place, we're going to have growth. We're going to have the interphase preparing the cell, and then we go into prophase, metaphase, anaphase, and telophase. All right, what have we got here? We've got the nuclear envelope. We have the chromatin network, which looks like spaghetti. We have the nucleolus, and remember, people, the nucleolus is for protein synthesis. All right, that is its function, and you learned that when we did all the organelles, structure versus function inside a cell. Then we have the centrioles. Now remember, the centrioles, there are two of them, and the centrioles are going to now split up when we have mitosis occurring. This little one here is incorrect. It shouldn't be there. All right, so that line should actually go all the way here to that one. So we've got two centrioles, and it makes a centrosome, the two together. Then we have the cytoplasm in the cell, and then the cell membrane. Now, you've got to understand that when we draw this, we are looking at what? The nucleus, because we're not going to worry about drawing the cell membrane and blah, 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 and all the other organelles. We are focusing only on the nucleus, all right, because that's what's going to be doing this incredible job. Now we go to prophase. And prophase, if you recall, I told you that what happens here, the chromosomes, instead of looking like spaghetti, they now actually lose water, and they dehydrate a little bit, and they shrivel up. And when they shrivel up, they end up forming the X's, this little X shape. All right, so it's a duplicate. So you've got a duplication of the chromosomes. Why? Because that's what happened during interphase. The cell was getting ready with all its chemicals so that it can produce two identical chromosomes. So when it opens up and separates out, we end up with two identical daughter cells, not son cells, daughter cells. Okay, so here we go. We have the asters. These are the little spindle fibers. I'm going to put in brackets here the spindle fibers. All right, so they form little asters. They're starting to grow. The centrioles, there are two of them here. And what they do is the one's going to start moving in that direction, and the other one stays at the top. All right, now, the nuclear envelope is still there, and it now starts to take over the whole main part of the cell. You've got the nucleolus starts to disintegrate. We don't need the nucleolus, all right? Why? We're not protein synthesizing now. The cell wants to make another duplicate cell. So there's no nonsense about protein synthesis and wada and all the other functions that are going on in the cell. All we want now is for this cell to now make a duplicate copy of itself. Do you recall I said to you, if for example I lose a little lip cell here, your lips get cracked, or you cut your finger, or you teenagers, you squeeze a pimple, what happens? You want cheek cells to replace that spot. You want lip cells to replace the spot on your lips. You want liver cells to replace cells in your liver. You certainly don't want cells from other places. So, how does that happen? It's when the cells next to the damaged area duplicate so that they grow over the damaged area and repair it. Okay, so nucleolus, no protein synthesis. It's going to start disintegrating, and you can see here it's starting to disintegrate. Then we have our pairs of chromosomes and the centromere. Now remember, the centromere is this little thing in the middle that looks like a little circle that literally joins the two chromosomes. All right, so it's joining them. That's your centromere. It joins the two chromatids. It joins them together. All right, so that is prophase. We're getting ready now. The chromosomes are ready, and they're going to start moving to the middle. And the middle is your next phase, which is metaphase. Remember, it's ipmat, interphase, not a phase, prophase. We're getting ready. Metaphase, everything's in the middle. So here we go. And this is where we stopped last week. Okay? Meta, remember, means middle. So it's easy to remember. Number one, it's in the middle of interphase, prophase, metaphase, anaphase, telophase. There it's in the middle, number one. Number two, look at this. Your chromosomes are all lined up at the equator. All right. They're at the equator, they're in the middle, and the centromeres are right there, and you've got your spindle fibers. Now we can go one further, and the spindle fibers that attach to a chromosome, okay, those spindle fibers are called, guess what? Spindle fibers, they're attached to chromosomes. Now this is hard, this is really hard, people. It's called a chromosomal spindle fiber. I mean, how hard is that? This is easy, as long as you understand it. So, not hard. It is the chromosomal spindle fiber. Easy as that. All right. Now, the ones, so you've got your hang on, what did I want? Just remember, your chromosomes are always equal numbers. Okay? Whenever you look at mitosis or at meiosis when you get to grade 12, you will always have an even number of chromosomes. Why? Because chromosomes come in pairs. One came from the mother and the one came from the father. So one set of chromosomes are going to be maternal, they came from mommy, or the female, and paternal chromosomes came from the father, okay, or the male. And that applies to plants, it applies to animals, all right, although I don't know so much if plants talk about the mommy oak tree and the daddy oak tree, but you know what I mean. All right, so, or in fact it would be cherry trees because oak trees all have one sex. But people, what I'm trying to explain to you is male, female. So the offspring, the zygote that gets formed, all right, when reproduction takes place, we are going to have one gamete with chromosomes from the mom, from the female, one set of chromosomes from the male, and they are going to join together during fertilization to form a diploid zygote. You are a diploid zygote. And so is the peach pip. from the peach you had for lunch this afternoon, or the apple pip from the apple that you had for lunch this afternoon. Okay? In there, you're going to have, it forms a zygote, and then what happens? It starts to grow and form an embryo. And your little seeds that you're eating are your embryos. Where did they come from? What do they have? One set of chromosomes from mom, one from dad. So there is always two. And if there are always two, there's always a pair. It's like wearing socks. Okay? If you have two legs, you need two socks to make one pair of socks. And people normally, I mean, conservative people like myself and John maybe, okay, we are going to wear the same socks. So if we wear socks, we're going to have two identical black socks and two identical white socks and two identical pink socks maybe, but they will be the same. We're not going to wear one pair of socks that's pink with red spots and another pair that's black. They must be the same. We call those homologous chromosomes. One from mom, one from dad, together, okay? They're in pairs. Here are our pairs. You see there's pair 1, and there is pair 2. All right? Belinda's telling me, I can't see. 2. So those two are from the same, and for the same characteristics, and those two are for the same, 1 and 1 are for the same characteristics. All right, now have a look at what they've done. They've lined up along the equator. So they're now sitting there ready and waiting for things to happen. So let's move on. Okay, we now go to anaphase. So we've done interphase, we've done prophase, we've done metaphase where everything's in the middle. We now go on to the A for anaphase. All right, people, we've got 2 minutes, so before we finish off, I just want to do anaphase. Now, how do you tell the difference between a prophase and anaphase? Prophase, your chromosomes are not in pieces, they're still in an X shape, and it's as simple as that. That's how you remember it. They're still in an X shape, that's number 1, all right? And number 2, they are random. They're all over the show. They're not together, they're not apart, they're just sort of wherever they want to be. Whereas look at anaphase. Anaphase is everything is in order. They're pulling to the poles. These spindle fibers are pulling the chromatids, people. They pull them apart, attached to the centromeres, okay, the little round goodies there, and they pull them to the poles. And here, we are also going to have, it's random. As long as there's one from mom and one from, it doesn't matter because they are all identical. Let's say this one came from mom, that one, let's get another color here. This one came from mom, that one came from dad. They're all pulling together, but they'll be identical to this one and that one. You follow? They're all identical. So that is exactly the same as that, and they're now pulling apart. But if you look at anaphase, they are organized. All right, I'm going to hand you over to John.
Great, guys. I hope you're getting this. Mitosis is really important, and get your questions. We're compiling the questions, putting them together. The last little while of the show, we'll take your questions. We'll make sure we get them all answered. So let's go to a quick break, and after that, we'll complete, and I'm sure we're going on from anaphase. What's the next phase? Telophase. Am I right?
Cool.
That's it. Let's do it. Go to the break now, and we'll see you just now. Welcome back, Mindsetters. We're doing Learn Extra Live, and it's Life Sciences today. We're here with Kathy, and we're going great guns. We're on to cell division or cell multiplication. The process we're talking about is mitosis. So get your questions in. Any questions that you have about mitosis, post them on the Facebook page. You know what the address is. It's www.facebook.com/Learn Extra with an X. We'd love to hear from you. Get chatting, get your questions in, and we'll see you, and we'll take those questions in after the next break. So let's wrap up the last part of the process of mitosis. Over to you, Kathy.
Okay, hi boys and girls, welcome back. All right, just to recap, anaphase what do we have? Everything is organized. In comparison to prophase, where your chromosomes were all scattered around the nucleus. Okay. Also something else is your centrioles are still together during prophase, especially early prophase. Here, they're at opposite ends, and you must be able to tell the differences, all right? Remember, chromosomes pull apart. Something I just also want to bring to your attention here. Do you see right here, everything is lined up? And as soon as those chromosomes now start to, or the chromatids separate out, and the centromere splits, as soon as that happens, then we say that we are undergoing karyokinesis. And karyokinesis means the splitting of the nucleus. Okay, so that's when the whole thing actually starts to happen, is when that centromere now pulls apart, okay, and the chromosomes are being pulled by the spindle fibers to the poles. But as soon as that happens, karyokinesis has started. All right, we've done anaphase, and here you can clearly see at this point that we're now moving, the chromosomes are moving completely apart, which means karyokinesis has definitely taken place. And you must know that term. Karyo means the nucleus, of nucleus, with nucleus. So karyokinesis, splitting of, breaking up of, moving of the nucleus. All right, so karyokinesis at metaphase. Anaphase, those chromosomes pull apart, and now we get to telophase. Some textbooks write it T-E-L-E. All right, so it's a difference between English and American. English, listen to me. Oxford English and American English, or British English and American English, both terms are correct. Okay, so telophase, what we've got our centrioles now pair up again. They appear in each of the chromosomes, where the nucleus arrives again miraculously. The chromosomes lose water and stretch out nicely like the spaghetti. Okay, the nuclear envelope is perfectly in place for each one, and we end up with something called an invagination. Now that's a very, very, very long word, but what invagination means is an indentation. So we end up with this little curve here, and the little curve's going to get deeper and deeper as the cytoplasm moves apart, and the nuclear membrane then does this. Okay, so that this becomes one structure, and this becomes the other structure. All right, so that's your invagination, and when this invagination or indentation starts, we say cytokinesis is beginning. Cyto meaning the cytoplasm, kinesis, the moving of the cytoplasm. All right, so cytokinesis is during telophase, and during metaphase, we have karyokinesis. Alrighty, people, now we are ready to rock and roll. What I'm showing you here is just everything in pretty colored pictures so that you can actually see the process here. We're going to have this. Can we see anything? No. We can see the centriole and the asters developing in this one here. All right. So if we just look at the board closely, there are the asters, there are the centrioles, the chromosomes are all just one big mush, all right? Here, and this would be, sorry, let me just write here, this would be interphase. It's the longest phase, and it's the preparation phase. Then we move on here to this little one here, so we'll make that 1, we'll make this 2. Over here are chromosomes, easily identified. Are they ordered? No, they're all mushy and randomly placed. Are they in pairs? Oh yes, they're in pairs, which means what? That this is a perfect little cell. All right? Everything here is in a pair. So all your somatic cells in your body, in fact, all your chromosomes as you stand there now, will be diploid. And if they are diploid, there was one chromosome from mom, one from dad, and that makes your chromosome pair. All right, so there they go. So this second phase here is prophase. All right, and we are now getting ready. Everything is happening nicely. After prophase, well, this here, if you look at this, your spindles have now formed, and this we would classify as late prophase. Okay, why? Because look where our centrioles are. They're sitting right at the ends of the poles, so they are here, and your spindles are now pulling across. Now we move on to the next phase, and this one we go interphase, prophase. And where is everything? In the middle. So this is number 3, which is metaphase. Oh man. There we go. Metaphase. All right, so there's metaphase. Why? Everything is in the middle. The chromosomes are beautifully arranged along the equator. So what does this tell you? Karyokinesis is about to take place. All right, now, easy peasy, this is phase 4, and that is going to be anaphase. How do we know it's anaphase? Look how beautifully those little chromosomes have now opened up. Here, and this is to help you identify it in an exam. And please don't give this as a reason. I think your teacher's toes will curl. But look here. Here your chromosomes look like butterflies. Okay? Here they look like little butterflies. They're very pretty. You know what a butterfly looks like. Goes. All right? So there they look like little butterflies. When we go into anaphase, what do we have? They look like little moths. Okay, they don't look very spectacular. So there we go. And that's how you're going to tell the difference. Metaphase, also everything in the middle. Anaphase, the little moths are pulling towards the poles. And then clearly the 5th phase, real phase, actually the 4th real phase, but the 5th phase is telophase. And your telophase, look how beautifully everything is here. You've got this invagination starting, and that means that we are going to have cytokinesis taking place, which is the cytoplasm separating out. And here, this would be late telophase. And if they gave you this in an exam, it would just be because they want you to use your common sense. Right. Early prophase, late prophase. Early metaphase, late metaphase, they've already started to pull apart, but they're still there in the middle. All right. Anaphase, now we have late telophase. Why? Because this imagination is much deeper. But look here, let me just get a different color. Look over here. Your chromosomes are beautifully in place. They're nicely in place in the nucleus. The nuclear envelope is perfectly in place. And we now almost, for all intents purposes, have 2 separate cells. So that's now going to move on to our 2 people, and this is important. 2 identical daughter. Identical daughter cells. Okay, and there are 2 of them, and they must be identical. Why must they be identical? Because you don't want a little heart cell, a cardiac cell bouncing around you on your face. All right, and you certainly don't need a liver cell in your eyeball. So we must replace the cells that are damaged, and that's it. Those that are damaged here, we must replace with the cells that are identical right next to it. All right, so 2 identical daughter cells. Okay, I promised you I'd show you pictures, and this is something teachers love to give their learners. Okay, so take a very, very careful look. I'm going to get a different pen, and my producer is going to focus in exactly where I want them on the board. So I'm going to draw on the board for you now. Okay, if you look at all these cells, this is from the tip of a root. So this here, can you see anything different? This cell, that one, they all look the same. One would say that they look normal. And the fact that they look normal, we are going to tell you that A is going to be interphase. Okay, those cells are preparing to divide. All right, I'm looking for the next one. Okay, let me see where my chromosomes are very nicely spaced out. That's C and C, B, D. All right. D is easy, we can identify, because look, what have we got? We've got a nice fat little nucleus there and a nice fat little nucleus there, which tells us that this cell is now dividing. It's in its final phase, so D would be telophase. All right? Unfortunately, we can't see a lot of detail, but let's have a look. Okay. For C, we've got a C there and a C here. Now, if you look at this one, I would have said that this is either late metaphase, so it's late metaphase, because if you look at this, those chromosomes are still in the middle. So if they're in the middle and they lined up here around the middle, it is metaphase, but they're starting to pull apart, so it's late metaphase. Just trying to see if we haven't got another metaphase here. All right, and then if we look at these ones here, we can identify the chromosomes very nicely, but they haven't really started to pull anywhere, so I would say that these two, so B, and I'm going to write across here, those two would be, you've got the chromosomes, you can identify them, that would be prophase. So we have interphase, we have prophase, we have metaphase, and there, that's clearly anaphase, because look at your chromosomes, they're all sitting here at the poles, so this would be anaphase. All right, and then telophase, where it pulls out. And that's what they're going to show you. And they're going to say, right, what happens in each phase? How did you identify it? Remember, interphase, you can see nothing. It's the longest phase. It's the prep phase. It's the chemical preparation phase. Then prophase, chromosomes are there. You can see them, all right, but they random all around the inside of that nucleus. And your centrioles are still together. Esters have started to develop. Then you look at metaphase, easy peasy. Metaphase, everything's lined in the middle and the chromosomes look like butterflies. All right? 2 chromatids held in the middle by a centromere. Then we move into anaphase, looks like little moths. Centromeres have split and the chromosomes are pulling apart. So karyokinesis has taken place. And then move to the poles. Now we have telophase, the invagination splits into 2 identical daughter cells. Very important. 2 identical daughter cells is in telophase. And the fact that the cytoplasm divides tells you it is cytokinesis. Okay? We've got like a minute left, so I just want to show you this. Here's prophase in it's magnified 2,000. I mean, look at this, people. Magnification 2,700 times. That is huge. And we call this a micrograph. But you would have learned about micrographs in grade 9. But you've got a 2,700 times magnification. We're looking at prophase. Can you see? You can actually identify the chromosomes, and they are very dark. They stained very dark. Then we go metaphase. Whoa, look here. Everything is in the middle. Remember, always in the middle is metaphase. Meta means middle. Then, oops, no man, I'm being...
You need to go to the top, Kathy.
Okay,
oh, no,
we're
there you go. You've got it. You've got it.
Metaphase, and we move to our next one, and there's metaphase. This is not metaphase. John made a mistake.
I made a mistake.
John does these for us.
He
puts all these slides on. He's quite a buff here, I have to tell you. All right, so this is actually John. Anaphase. I hope that you know this whole process now, huh?
Yeah, I do.
All right, so anaphase, why? They're pulling towards the poles. What are? The chromatids. And then we have telophase, which is the end phase, and everything's pulling out nicely. We've got our two cells here. There's the one, there's the other one. They are identical, and there are two of them, all right? And what's happened here? Last thing that you must remember is cytokinesis. And if you don't get full marks for what we've done today, whoa, then I really don't know. Over to you, John.
Well, thank you, Kathy. There have been lots of questions on the page, and I'm sure that you're getting the stages right, making sure that you've identified the features. Now, one of the questions that we had was, somebody said, but our textbook diagram doesn't look like that. That doesn't matter. You need to be able to see the feature and make sure that you can identify what's happening. So like we know, metaphase, even I can get it right, it's in the middle. Make sure that you've got that. Telophase, where you get the invagination. So make sure that you pick up different diagrams and that you follow these phases through, and make sure that you can see different slides. We've tried to show you a variety from a root tip, from a diagram, and we're going to go and have a look at a few more, but we're going to go to a short break, and we'll also then make sure that we answer some of your questions. So I'm sure that you're ready for just pick up a glass of water, and don't go too far away. We'll be right back after this very short break. Hi everyone, welcome back. We're doing Life Sciences Live this afternoon with Kathy, and we're looking at cell division, looking at mitosis. Now, make sure you've got the stages right and that you've got the diagrams all sorted. We're going to take a look at a couple more slides. These are important because you might not get the diagram that's in your textbook, so the more diagrams you see, the better. And then we'll answer some of your questions that are coming in. If you've got important questions, urgent questions, please post them now onto Facebook. We'd love to hear from you. But before we do that, let's go to Kathy with those last few diagrams on mitosis.
Okay, people, there seems to be some confusion about mitosis and meiosis. Meiosis is for sexual reproduction. Mitosis is for growth, repair, replacement, and reproduction in unicellular organisms. I'm going to do that and just show you very simple diagrams to remember it by. Clearly there's some grade 12 learners watching this. Okay, and you need to know what mitosis is anyway, grade 12s. All right, here we're looking at an allium root tip, and you have, now remember, they can give this to you in an exam as a photo. All right, so make sure that you actually know and you can recognize from micrographs, which are photo impressions of very, very, very tiny things that you saw on an electron microscope. Okay, so interphase, again, it's the between phase. The cell looks normal. There doesn't look like any changes. Everything is chemical that's taking place. The cell is in preparation. All right, it is prepping to divide. Prophase. You can identify the chromosomes. They've started to thicken out, as you can see there. In this case, we can still see here the centriole, all right, I mean the nucleolus, which means it's still early prophase. It's just started to be prophase. If we look at this one here, prophase, your chromosomes are now starting to separate out nicely. Prophase has occurred. Then we have metaphase. Everything's lined up in the middle. And remember, we're going to have our chromosomes looking like little butterflies. So you've got your two chromatids joined by a centromere. So just to recap, those are chromatids. All right? And there is your centromere. Okay? And now we're going to anaphase. How do we know? This one's a bit difficult to see that it's anaphase, but if you, and I'm going to use, let's use blue. If you look here, look at the shape of the chromosome. So this would be really early anaphase. It's very early anaphase. Here, you're going to find that karyokinesis has only just taken place. All right? Then we end up with our anaphase. This is an easy one. I mean, if you can't tell me that that's anaphase, I really think I've wasted 2 hours. All right? Which I know I haven't. I know you can do this with your eyes shut. And then telophase. I mean, whoa, look at that. And how do we know this is a plant? Because you don't have an invagination developing like you have with an animal cell. All right? What are you going to have? You're going to have a cell plate starts to grow across like that. And as this cell plate develops, so it'll go all the way across and cytokinesis will take place, except that this is a plant cell. And plant cells and animal cells, you know, plant cells have got that cell wall, and that's a bit of a problem. So we've got to have this cell plate that grows across. And also, people, make sure that you know the difference between an animal cell and a plant cell. If you're looking at mitosis, how are you going to tell the difference? Number one, you look at telophase, at this invagination, or the cell plate, like I've just told you now. Also, your plant cell is not going to have centrioles. Why? Because you only find centrioles in animal cells, and you should know that from your comparison of animal and plant cells. Alrighty, and then if we look at telophase here, this one, there's your cell plate, and the cell plate has now developed all the way along here. Okay, and we then end up with our two identical daughter cells. They are identical, right? And that's us. So what I want to do here is just get myself some space to write. Okay. The difference between meiosis and mitosis. The grade 10 learners, you must know mitosis. For the grade 12 learners who are posting questions, you're now getting confused between meiosis 1, meiosis 2, because you are looking at meiosis. We are doing mitosis. All right, so we do mitosis, and this is for growth, repair, replacement, and reproduction in unicellular organisms. All right, and what are we going to have? We have one cell which is diploid. It's got a full set of chromosomes. All right, a full set of chromosomes. It's complete. It's got one set from dad, one set from mom. N plus N makes it 2N. And this cell is now going to make or copy, make a duplicate, a perfect duplicate of itself. Two identical daughter cells result. That's mitosis. For meiosis, and let's pick another color. We're going to do meiosis in pink. Okay? This is meiosis. Now listen to the word. Meiosis is for sexual reproduction. Now what happens when there's sexual reproduction? We have an egg. We have sperm. We join the two together, and voila, we have a new little offspring. Okay, the offspring is like you, like mom, like dad. But if we're going to have dad and mom, we can't have 2N plus 2N, because that's going to give us 4n. So what must we do? We're going to say, no, 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 in the reproductive structures. So in the anther of a plant, and the ovary of a plant, a flower, and anther and the ovary of a flower, and the ovaries in an animal, and what do we have in the male? Little testicles that are going to produce the sperm. There we are going to have meiosis taking place. Why? We have to take the 2n from mom, and we're going to go, what's going to happen? This 2n has to now divide into n plus n. And then we are that's meiosis 1. And then we're going to duplicate those so that they are the same. And that's meiosis 2. And then we're going to do this. So what do we end up with? 4 unidentical daughter cells. If this happens in the ovary, you know what? One of them is slightly bigger when that division happens. And we're going to have one slightly bigger here, and then these little ones. And these little ones will all go to make food and provide nutrition for this little one here. Okay, if we look at meiosis in a male, let's do a different color. For meiosis in a male, we start off with 2n, and in the testicles, we are going to have reduction division into 2 gametes, but remember, the male is XY. So we'll have X and Y. Then these duplicate, and these duplicate. So this part here is very similar to mitosis. But what are we doing? We're duplicating. Are they the same? No. Half of them are X and half of them are Y. Okay, so mitosis, 2 identical daughter cells that are haploid. Meiosis, sexual reproduction. We have to take 2n, diploid. We've got to halve it. So when we take half from the male, half from the female, we join it, what are we going to end up with? A perfect 2n again. Just quickly, I've got 1 more minute, and one of the questions that came up as well was on cancer. Cancer is a form of mitosis. What happens is, one little cell undergoes mitosis, and it's a little bit wrong. There's a bit of a mess up in that mitosis process. So the little cell says, oh dear, the one that results, it says, I'm not right. I'm going to make more so that I can make sure that I can fix it, except it never does. And when it grows very fast and it forms a benign tumor, then we can cut it out. But when it is, come John, help me.
Malignant.
Malignant, okay. Malignant means that that tumor sits there and the little angry cells, just think of them as angry cells. They're Lincoln Park listeners. Okay, they break off and they cruise around the body, and they say wherever they get, they start making more cells. And that's when it is malignant and it spreads all over the body, and it's bad news. All right, generally breast cancer is you start off with a benign tumor, and when we don't look after it, it then becomes malignant. To John, here you go, people. I hope I've helped you. Have an awesome week till next Wednesday.
Thanks, Kathy. It's really been a great lesson. Make sure, grade 10s, that you don't get confused now. You only need to worry about mitosis. It's the grade 12s that are getting confused. Meiosis comes in grade 12. Am I right, Kathy? Yeah, okay, cool. Now make sure that you've got those steps. Practice, 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 and all the best for the coming week. Go to the page. If you've got any questions, the help desk will also help you out. And we'd love to see you again next Wednesday afternoon. From us, Kathy and myself, bye.